All right, so let's get started. Like I said, this presentation does not provide any medical advice. The health-related information is for educational purposes only. It's not intended to be a substitute for medical advice or treatment or diagnosis or anything by me. So you should consult with your doctor or your practitioner when you incorporate any of the educational material that you learn here today from me with your doctor or practitioner. And when you want, if you want to incorporate any of this into your wellness plan. And it's your responsibility as a participant to research and validate any of the information that I provide in this presentation with peer-reviewed research. And the participants of this course are encouraged to obtain original copies cited through either open access, if available, or through purchase through appropriate sources of your references that you look for. Okay, so let's get started. Is this you? I don't feel well and I'm confused. I know the immune system is involved, but I don't understand it. So it all starts where in the with the mitochondria function. This is where the energy production takes place. But it also affects the immune regulation, histamine regulation, adrenal hormones, the HPA axis, and the limbic system, which is the processing of external stimuli and converting it into chemical signals to give instructions to the body on how to respond, and it affects the brain function. So let's start from the beginning on how energy is made and how it's connected to the immune system. This is a basic diagram showing that fats, protein, and carbohydrates ultimately make ATP, which is the energy source that our body uses. ATP for our body is like gasoline is for our car. We can't just pull the oil out of the ground and put it in our car and expect it to run. We have to send it to the refinery. And I'm sure you've all seen pictures of the elaborate machinery needed to make gasoline. Well, our digestive system is even more complex. So once these foods are broken down into small molecules, the nutrients are then absorbed by the body and used to make energy for us. That energy is called ATP. This process happens in most organelle that we call the mitochondria, named the powerhouse of the cell. And this process can be affected by many factors, and there's a lot of opportunity for mitochondria function and energy production. So what do the mitochondria do if it detects a foreign substance such as a pathogen or a toxin? Well, the mitochondria on the surface, they have proteins on it that sense a voltage chain that tell the mitochondria how to respond. The mitochondria will respond the same way that we respond when we are attacked. And we stop communicating with the attacker, we throw up walls, and we withdraw our support. The mitochondria are the gatekeepers of metabolism. If something tries to steal energy from them, they react very quickly by initiating a cascade of changes that change the structure and function to minimize the energy steal. This is an evolutionary benefit in order to help the cell survive a hostile. They will change their lipid bilayer structured membrane and make it thicker so it's less permeable. Then it reduces its use of oxygen in the electron transport chain. If you remember from biochemistry, oxygen is the final acceptor of electrons that make water and ATP. With toxic illnesses, our immune system becomes dysregulated, and many people get stuck in the inflammatory side of the healing process and are unable to complete the full cycle to resolution. With mitochondria leaking ATP and oxygen, we have more extracellular oxygen available to make reactive oxidative species, which is free radicals to help kill pathogens, which is, however, with excessive mitochondrial leakage, there will be more stress signals preventing it from completing the healing cycle. The stress chemistry will also use that ATP in the production of neurotransmitters to aid in the fight or flight response. So the result of this is the ATP and the oxygen leaks out, and it results in fatigue, weight gain, inflammation, and neuronal damage. So the mitochondria activates the immune response. I'm sorry, people keep coming in and I have to click the admit button. Anyway, so all cells will leak ATP. Okay? And ATP outside the cell is going to signal to surrounding cells that there's a trouble. This triggers a cascade of surrounding cells to follow suit of protection of their mitochondrion, and they will also decrease their ATP production as well. And this leads to increased ROS, which is oxidative stress. Now, our mitochondria spend their life cycling through the 4 phases of the cell danger response cycle. So here we have a normal working mitochondria, and the normal healthy mitochondria are involved in redox reactions, transferring electrons from hydrogen and oxygen into water while producing ATP to provide the energy for activity through glycolysis. Physical activity helps stimulate a normal sleep-wake circadian rhythm, and this cycle will produce autophagy, non-inflammatory cell repair and recycling. Autophagy is the term used to describe the healthy debris cleanup and repair of old damaged cells. Then we go to the cell danger response. Phase 1, which is inflammation. Once the proteins on the surface of the mitochondria sense a voltage change, it's interpreted as, oh no, something is stealing energy. And during the stress, the mitochondria will fragment within 5 minutes, and ATP and oxygen is pumped out. It will decrease its use for oxygen, which will lower ATP production. And the citric acid cycle will shut down shifting pyruvate to make lactate instead. And this buildup of lactate causes a lot of pain. And it will shift from a redox metabolism state that's used in the citric acid cycle towards using glycolysis. And this is the self-protective mode that is pro-inflammatory, which is necessary to stimulate immunity to kill a pathogen threat. Then we move over to the cell danger response 2, phase 2, which is the proliferation stage. The triggers of the danger have been addressed and removed, and the mitochondria are no longer receiving signals of voltage changes. Now it's time for the mitochondria to bring in the nutrients needed, such as amino acids, B vitamins, and minerals, to restore its normal redox function and to maximize ATP production. This is the proliferation stage, which is very anti-inflammatory. Then we move over to the phase 3, which is the remodeling stage. Okay, so in this stage, lactate is going to go back to pyruvate, which can be used to fuel the citric acid cycle to produce electrons that ultimately go into the electron transport chain, and that uses oxygen as the final acceptor. The normal circadian rhythm in sleep is restorative, and you should wake up feeling rested and restored. And then we complete the whole cycle. Now, there are many factors that activate this cell danger response. And when the immune system becomes dysregulated and is unable to swing back in balance, and including advanced aging, infections, hypoxia, sleep, stress, vagal motor outflow, blood sugar dysregulation, traumatic brain injury, nutrient imbalances, diet and food intolerances, dysbiosis, genetics, hormones, toxins, and toxic mold exposure. So in my immune course for the practitioners, I go into depth into each of these factors and how they are related to immune dysfunction and how to fix it. Okay, so this diagram is a normal pattern of how the immune system works. The yellow line is increasing number of pathogens that are multiplying. You can see here that the yellow line goes up, and then it, as the green line goes up, which is the inflammatory and the immune ramping up to kill the infection, then the infection becomes resolved. And then the pink line is how we resolve inflammation in the immune response. So the immune suppression goes up, and then it resolves the inflammatory and in the immune response to that pathogen. Now this is low. This pattern is low immune suppression, which promotes a high sustained inflammation. So you can see here that the yellow line comes up, and then the green line comes up really high, and it stays high, okay? And because the pink line, the immune suppression, doesn't come up high enough to resolve the inflammation. So you basically have chronic inflammation without resolution. And it's not resolving because we don't have enough of the pro-resolving mediators, which is EPA and DHA, that prevents resolution. And that comes from, and the resolvins, they come from fish oil, okay? So fish oil is very useful in resolving inflammation. And they're converted to prostaglandin E2, which is very anti-inflammatory. And PGE2 is blocked by NSAIDs. You know, those aspirin, ibuprofen, those are NSAIDs. So in the long run, those NSAIDs will block resolution. So in essence, NSAIDs are toxic. And without the resolution, you get fibrosis and scarring. Okay, and here's the third pattern. In this scenario, the immune response is insufficient to resolve the infection and inflammation. And so we'll get into some of those reasons later, in the later modules in the immune course that I'm making. But you can see here's the infection with the yellow line, and then the immune response comes up, but it doesn't come up quite enough to resolve the infection. And then you have the immune suppression coming up. So what happens is it results in a low-level smoldering infection and chronic inflammation. And this is very common scenario with mold toxicity and other toxic conditions, and also in autoimmunity. So the immune system becomes chronically low-grade suppressed, allowing for ongoing inflammation and infections such as viruses and Lyme-like bacteria. So now you're chronically inflamed and you're chronically infected. This is a very common scenario with body toxicity. The immune system becomes chronically low-grade and suppressed, allowing for ongoing inflammation and infections such as viruses and Lyme-like bacteria. So in the last two slides, inflammation and/or infections have failed to resolve because there's a lack of resolvins. And the resolvins come from EPA and DHA, which is from fish oil, and which is very useful in resolving the inflammation. To make resolvins, the EPA and DHA are converted to prostaglandin E2, which is anti-inflammatory. And NSAIDs block the PGE2, and they are resolution toxic, and you end up with fibrosis and scarring. So let's talk about the body's lines of defense. Our body has several lines of defense. First are the barriers, okay? Externally, the skin, and internally, the mucous membranes, which is the lining of the guts, sinuses, and lungs and bladder. If those barriers are breached, then the two arms of the immune system are activated. First, the innate immune system are activated, which are non-specific ground troops, like an Army infantry platoon. Then the adaptive immune cells are activated, which are targeted specialized forces, more like the Marines. These will be discussed in detail later on. And then we have the adaptive immunity, which is antibodies, chemokines, and cytokines. So the skin is our first line of defense. You know, externally, it works as a physical barrier. It has a low pH to discourage pathogen growth, and it contains normal flora to help crowd out the space available for other bacteria on the skin. But sometimes the skin is damaged, and this barrier is breached. And the invaders, such as bacteria, virus, and fungi, also the allergens called antigens enter the body. So the damaged skin, it allows intruders called antigens into the bloodstream. And the antigens can include chemicals, bacteria, virus, fungi, parasites, and pollen. And this stimulates the immune system to mount a response. This can include all of these things that I just listed here. So we have a large portion of our immune system in the lining of the lungs and the gut and the sinuses. And then we have stomach acid that acts as a barrier by killing pathogens on our food. Once one of these barriers have been breached, then our immune system steps up to the plate as the second line of defense. To understand what happens next, let's review the basics of the immune system. So the innate immune system, we have the inflammatory response that's designed to attract cells and substances to fight the intruder. And we have the phagocytes, which are various types of white blood cells that gobble up non-specific foreign particles. And we have the cells of the innate immune system that include neutrophils, macrophages, and dendritic cells. Now, to make immune cells, it all starts with a stem cell in the bone marrow. Most of the cells develop in the bone marrow, but some also develop in the blood and in the skin into various types of immune cells. Most of them will travel from the bone marrow to the gut. So you'll find the majority of your immune cells there, about 60 to 80%, because that's where most of the antigens enter our body through our food. So what type of immune cells do we find at the gut lining? We have the macrophages, the neutrophils, and the dendrites, okay, the dendritic cells. They sample the food that come in and the air that we breathe. And the macrophage, the big eater, will eat anything that isn't recognized as soft tissue. Then it secretes enzymes that liquefy and degrade the pathogen, destroying it. And the macrophages aren't really specific in the type of pathogen or toxin it engulfs. We have special immune cells that do that. And then we have mast cells that have granules on the surface and is where we store histamine. If these granules get knocked off, then histamine can be released. And then we have lymphoid cells, which can develop into antibodies of the immune system. So the immune system samples everything that enters the body to determine if it's a part of our body or not. If it's not part of self, then it just floats away, no harm done. If it's not self or antigen, then the dendritic cells and the macrophages go into action. In this diagram, you can see the green dendritic cell reaching up between the epithelial cells in the GI tract to sample the bacteria in the lumen or the center of the GI tract. This sampling is done by dendritic cells and macrophages. These immune cells grab a hold of particles and pull it down into the tissue. And pathogens in the gut that have not been killed by gastric acid in the stomach can be introduced here and enter the body in this manner. Usually the immune system recognizes them and gets rid of them. However, some are very virulent and cause disease. The myeloid-derived suppressor cells will develop in cases of, it causes lots of inflammation. And what happens is we have immature myeloid cells that are inside the bone, and then they normally they would mature outside the bone into neutrophils, macrophages, and dendritic cells. However, in the face of chronic infection and inflammation in a dysregulated immune system, we make myeloid-derived suppressor cells, which cause an impairment of the innate immune response, increased risk for infections, increased risk for autoimmunity, loss of the ability of immune surveillance, and it in fact it activates inflammasomes, which we'll talk about later. On the next slide. So inflammasomes are immune complexes in the cell. The inflammasomes are drivers of inflammation and the biggest driver of autoimmunity. A damaged cell will release their intracellular contents into the tissue matrix, and this causes more infiltration of inflammatory cytokines, and the contents spilled out will communicate to their surrounding cells to commit suicide, instead of a natural apoptosis and clearing of immune cells. So you now have a condition called pyroptosis, which is flaming cell death, instead of a natural apoptosis and clearing of immune cells, with a downward spiral of inflammation and immune activation. So metabolic stress will also activate inflammasomes, and they will end up releasing their intracellular components, which is very inflammatory. It signals surrounding cells to commit suicide. And you'll see this in conditions such as metabolic syndrome, insulin resistance, and type 2 diabetes and cardiovascular disease. These are examples of metabolic stress that perpetuate the inflammatory response. And inflammasomes are drivers of autoimmunity and inflammation. Rarely will you see autoimmune disease without the inflammasome activation first. Then you get the depletion of NAD, which is needed for mitochondrial function. And you might have heard me talk about NAD as being niacin that we need to make energy in the mitochondria. So what happens is you end up getting tissue hypoxia, and many times this is the low-hanging fruit. And this can happen with sleep apnea, COPD, cardiovascular disease. You know, people that when you travel to high altitudes, or if you have poor circulation and intermittent claudication and living a sedentary lifestyle, or you're just so sick you can't get up and move around, you're not getting enough oxygen to the cells. So the treatment is adequate clean air, oxygen, and circulation as the first line. Now let's talk about how the mitochondria and the cell danger response is triggered by antigens, and even those inflammasomes can end up causing so much damage. It's considered as an antigen in the body. And remember that all the foreign substances that come into the body are called antigens too. And this includes bacteria, virus, and fungi, as well as toxins from food additives and colorings, preservatives, and even the VOCs and pollutions that we breathe in, and even mycotoxins. So now you've got your mitochondria shutting down, triggering this, the immune activation. So let's go step by step. This is what happens. So you have the mitochondria cell danger response happening. So what happens is you get it decreases its energy production, and then it exports all the ATP and oxygen outside the cell. And then you increase the enzyme called NADPH oxidase. It's going to act on that oxygen and make reactive oxidative species, okay, which is very inflammatory. And that creates a lot of cellular debris. And the cellular debris will activate or stimulate the autoimmunity pathway. And it also decreases autophagy and repair. So now you're unable to clear the damaged cells and get rid of them. And that lowered ATP production decreases autophagy and repair. So then what happens next is you get an increase of the stimulation of the inflammatory immune response. And that causes increased neuronal stress. And now we have neural inflammation. And that leads to depression, behavior disorders, and systemic inflammation. And we also decrease our vagal motor outflow from the brain that regulates our digestion. So you lose your motility in your gut and end up with constipation. You lose your ability to stimulate enzymes to digest your food, and you lose your gastric acid secretion. And so now we have system-wide inflammation. And you can see why the mitochondria damage from whatever source can lead to neurologic and behavior disorders, along with many GI issues and chronic inflammation. So let's look at the basic response of the adaptive immune system, okay? So we have the B cells on the left, and they make antibodies. Those are extracellular. And then we have the T cells on the right, which make cytokines, and they're cellular. People keep jumping in this thing. Okay, so in their intracellular, they make cytokines, and they basically will go and kill the whole cell, okay? And both of these B cells and T cells are part of the adaptive immune system. Now, they kind of have some similarities because the B cells will make the antibodies, and IgM is the first made. And the T cells make an immature T cell, which is THO, and that's the first made. And the IgG is made in response to bacteria, fungus, and viruses. And the TH1 is made in response to bacteria, fungus, and virus. IgE is made in response to worms and allergies. And TH2 is made in response to worms and allergies. IgA is made in response to some infections and some food. And Tregs are made in response to food and infection also. And also TH17 is made in response to mold and autoimmunity. And TH9 is made in response to asthma. So the role of the innate immune system is to keep everything under control, while the adaptive immune system cranks up the cytokine production for a more specific response. Now, the B cells make antibody, okay? And the antibodies are part of the immune response that go after a specific antigen. It recognizes particular proteins on a surface, and it sends out an eat me signal once it binds to it. And once the antibody is bound to it, then the antigen loses its virulence, and it makes the pathogen much weaker. And each antibody is specific to an antigen. And once you've developed antibodies, then the next exposure of that to that same antigen will cause a cloning out of that antibody very quickly, and the immune response is much quicker. So you can typically snuff out an infection before it even starts. Now, the T cells are responsible for targeting and killing the infected cell and the pathogen once they gain entry inside of our body. And the T lymphocytes, they mature in the thymus, and these T cells make killer T cells, and they go around the body looking for cells that are infected, and they kill the whole cell, but they only recognize the proteins that are on the surface of these antigens. Now, the chemokines and cytokines are released by the immune system so that they can attract immune cells to the site of infection. And we know we see and feel this because it is accompanied by inflammation. It causes pain and heat and inflammation and also mucus. So the main reason for the mucus is to mechanically rid the body of the invader instead of bringing the substance into our body for processing. A mucus can prevent a pathogen from entering the body before it becomes a problem. So there's an evolutionary benefit for mucus formation. So the next thing you ask is, well, so how do we get this infection out of the cell or out of the bloodstream. So it can be done in two ways. The immune system has two arms. And here's the two arms of the immune system. And the battle takes place on either or both of these two. The innate immunity on the left, and then the adaptive immunity on the right. And you can see where they interface here in the middle. If you notice in this diagram, we already discussed the macrophages, the big eater, and the dendritic cell, and the mast cells, and the innate and lymphoid cells. And there are three that we didn't discuss yet, which are the basophils and the eosinophils and the neutrophils. And the basophils and the eosinophils are related to allergies, and the eosinophils more specific for worms and parasites. And the neutrophils are similar to macrophages, and I'll talk about that and more in a few slides later. So all of these phagocytes act on extracellular antigens. And the cells of the innate, the adaptive immune system is the CD4 T cell, and these are helper T cells. They make helper T cells, which differentiate to depending on what type of antigen. And the CD8 T cell, those are just kill. They just go directly after that pathogen, they just kill it. And then we have the B cells, and the B cells make antibodies, and which we also call the immunoglobulin. And there's this overlap right here in the middle with interferon gamma and natural killer T cells. And they're both T cells. Now, on the innate side, some of the properties is that we're born with this whole innate immune system, okay? We have it for life. It's not specific. It's the first responder. It has general housekeeping chores, okay? It's always patrolling, and it recognizes about 1,000 patterns. And the intracellular immunity, it's developed over time after we start eating solid foods, along with the development of the microbiome. We can still survive if we lost it, but maybe in a bubble. They're very specific, and it's a late responder, and it recognizes over about 10 million patterns. So the main purpose, the main job of the innate is basically to hold down the fort while the adaptive immune system revs up, so it can go in and make B and T cells to kill it. It may take 3 or 4 days to make all of these adaptive immune cells to get in there and kill, and maybe even up to 7 days to mount a full response. And these T or B cells, they'll go through clonal expansion, and they replicate logarithmically to about 10 to the 5th power to kill the pathogen. Okay, so let's talk about mast cells for a minute. You can see at the bottom left, you see a dendritic cell, and it's attached to a Th2 cell right here. You see a dendritic cell that's attached to the Th2. You see that the dendritic cell can directly release an allergen right here to go sit on the mast cell, okay? And there's a membrane-bound IgE. This is that Y structure there on the mast cell. And that can cause it to fall off when the antigen goes and sits on that IgE. And when it's released, then histamine, leukotrienes, platelet activating factor, IL-4 and IL-5 are released, okay? And so this is a way that allergies can tip over into a Th17, is by all of these things that are being released. Now, note, once the mast cell has this IgE, it can act immediately. It can just it doesn't have to go through all of these processes and go through the B cells and the T cells. It just releases things immediately, and it bypasses all of this requirement. Now, the Th cell is also releasing IL-4 and IL-5 right here, but it also releases neuropeptides down here too. And the histamine releasing factor, now the mast cell can lose its granules and release the contents of the histamine, lipids, and more cytokines. So you can see how the IL-5 here where is it, right here, can also stimulate the mast cell directly, which releases all of these leukotrienes and platelet activating factors. And the eosinophils release substances that cause brain fog and break down collagen. And this causes you to have increased aging of the skin when you have chronically elevated eosinophil. So you can see this creates a lot of interwoven loops. And once it gets going, it's self-perpetuating and you can get stuck in a mast cell crisis. So how does the immune system know when to secrete the T or B cells? Okay, so we have the B cells. It attacks an antigen outside of the cell, and like that's floating around in the body fluids. And then the antigen will react to any type of foreign invader, including chemicals and other toxins. And there's something called complement involvement, which I'll talk about complement in a minute, and that causes the killing of the bacteria and the cellular debris. And there's it induces a series of inflammatory responses to help to kill and fight the infection. And then the T cell, once the antigen gets inside the cell, then we have the T cell that gets stimulated. And usually it recognizes a protein on the surface, such as a bacteria, virus, and fungi. T cells don't really react to chemicals or pollution and stuff like that. It recognizes an invertebrate and like a protein surface. And there's a co-stimulation because the protein on the surface that activates the T cells have a receptor, and it's the CD8086. And then the cytokines of the T cell are released. Now, there's a crosstalk between T and B cells to destroy pathogens. And the cells of the innate immune system also work together with the cells of the adaptive immunity. And I'll show you how this works later. So a clinical pearl here is that when you block the co-stimulatory pathways, it will prevent the activation of the autoimmune pathway. Now, what will happen when a toxin enters the tissue is that a neutrophil from the innate immune system will act first. That's the little purple guy you see there. And they will enter the tissue and gobble up the antigen. Then the neutrophil commits suicide, which is also called apoptosis. And then the macrophage, the brown Pac-Man looking guy, comes in and along and engulfs the entire neutrophil. And then bits of the antigen will be placed on the surface of the T cell, which will respond by releasing killer T cells or by cranking up production of Th1, Th2 cytokines. If the cytokine IL-12 is released, a Th1 cell will be produced. If the cytokine IL-4 is released, then the Th2 cells will be produced. This will activate B cells to make antibody. Then the antibodies are very specific to that antigen and will go after the antigens that are outside of the floating around in the body. So what happens when an invader enters the body? So here we have the invaders in the tissue here. And when it comes in the tissue, it releases these chemicals, the TNF alpha, IL-1 beta, IL-6, and IL-8. These are released that cause blood vessels to become sticky. And this is going to cause the neutrophils to migrate down into the tissue, and they're going to gobble up the pathogen, okay? Then the monocytes, they're attracted to the tissue where they turn into macrophages, and then they engulf the neutrophil. Now the macrophages gets rid of the neutrophilic contents by taking it to the lymph to be eliminated. Then inflammatory chemicals are secreted, and that's prostaglandin E2, TGF-beta, and IL-10. So then the next step is that E and D series resolvins and neuroprotectins are secreted. The E series resolvins are from EPA, and the D series resolvins are from DHA. And these promote the pro-resolving mechanisms in the inflammatory process. Now the inflammatory process is resolved, and this is how it's supposed to work. Now, I want to introduce you to another important part of the immune system. It's called complement, because it's complementary to the innate immune cell functions. When the pathogen enters the body and passes the physical barrier systems, it's met by the innate immune system first. But the macrophages can't really eat the pathogen without help to bind to it first. This is where the complement immune fits into the grand scheme of things. So here's the complement proteins. And these are soluble proteins that are floating around in the bloodstream. And they're not active until they come into contact with a pathogen. That way, they don't start attacking your own self-tissue. The complement activation pathways are quite complex and beyond this course, but let's just say they work in tandem and interact with each other in a cascade of activations. These complement proteins do three things. Number one, optimization. Number two, form a membrane complex, attack complex. And number three is inflammation. So the goal is to kill the pathogen. So optimization means that the complement protein puts a sugar coating on the outer surface of the pathogen, and it makes it really tasty for the macrophages and phagocytes to engulf the complex easier. The macrophages have specific receptors for the complement proteins to bind. The attack complex will create a hole in the pathogen that causes it to break open and spill out its cellular contents, and this is called lysis. All of these pathways will result in making C3A and B. C3A causes inflammation and stimulation of the mast cells to release histamine. C3b causes opsonization and lysis of the cell. The new food inflammatory test that I'm using is called the Fit Food Test. It's a measure of this complement activation in the tissue. So these proteins, this membrane attack complex, this is very, very inflammatory in the tissue, and a lot of foods can get in the tissue and create these complexes. And the new test is actually measuring the inflammasomes and these C3s, these complement proteins that are forming to foods. And that's why these new that's why I switched over to using the Fit Food Test. So if you're having a lot of myalgias and gut symptoms, it's really a good idea to have one of these tests done. So what could possibly go wrong in the immune system? Well, several things can go wrong. If you have too many neutrophils entering the tissue at one time, then, and what happens when you got so many of them entering the tissue at one time and they become activated? You know, they only have a few hours before they undergo apoptosis, which is a natural cell death. Okay, if they're not cleared within a few hours by macrophages, they will undergo what we call secondary necrosis. And this causes a lot of tissue debris. They'll start spilling out their toxic contents into the tissue, creating more damage and recruitment of inflammatory mediators. So when inflammation resolves normally, white blood cells migrate into the tissue when they're needed, then cleared out through the lymph. When there's damage, you either have what we call DAMPs or PAMPs. And DAMPs is a damaged associated molecular pattern. And when there's damage, there's fragments of tissue. And PAMPs is a pathogen associated molecular pattern. And there's markers on the surface that tells your immune system that it's an invertebrate, such as a bacteria, virus, or fungus. So another scenario is, what if we don't have enough monocytes to clear the neutrophils? okay? Normally, the neutrophils undergo apoptosis once it eats the antigen, and a macrophage comes along and clears it by taking it to the lymph for safe disposal. However, if there aren't enough macrophages to clear the neutrophils within a certain length of time, the neutrophils undergo secondary necrosis. And the stress chemistry released from like toxins and even mold exposure can inhibit the macrophage production too. So you got a lot of inflammation going on, and you're getting your macrophages suppressed. So now your innate immune system isn't making macrophages. So even if you have a natural amount or normal amount of neutrophils, you're unable to get rid of them, and then they end up causing a secondary necrosis. So they only have about 6 to 10 hour window because they have a lifespan of their half-life is 6 to 10 hours. In that time, 50% of the neutrophils are going to die by then. And if they're not cleared in that time frame, they'll undergo secondary necrosis, spilling out their toxic contents. That's going to cause further tissue damage and more recruitment of neutrophils and more inflammatory cytokines. So we have two scenarios. The neutrophils enter the tissue quickly because there's lots of antigen. Okay, and then the other scenario is that we have poor functioning innate immune system. We have the underproduction of monocytes, and this results in a chronic infiltration of neutrophils. This is the primary mechanism of the initiation of the autoimmune disease. Now, anything that feeds the inflammatory process will feed into this autoimmune process. Now, let's talk about neutrophil necrosis a little more in depth. So ATP and IL-1 beta are intracellular molecules, and they're not supposed to be like floating around in the cytosol, okay? They're supposed to be inside the cell, but when the cell, when like the neutrophil dies, this is spilled out. And when the neutrophil dies from necrosis, if the neutrophil undergoes apoptosis, which is natural cell death, it doesn't like spill out. They're all contained, and then the macrophage comes and gobbles it up, and then that way it doesn't cause so much damage. But when these two things are spilled out, it's basically a signal to the surrounding cells that to commit suicide as well. And so then they release their debris to form from a necrotic neutrophil, causes a lot more infiltration, more tissue damage, and more neutrophil cell death. And then we have these nasty enzymes that are supposed to kill the pathogen that's inside the neutrophil called performs and granzymes, and they are cytotoxic. They're used to bind to that pathogen and create large pores in the cell of the pathogen to kill it. But when they're released out into the out of the neutrophil, it damages our own tissue. Now the neutrophils secrete something called extracellular matrix and metalloproteinase, which we just call MMP9, and these can be measured in a test. And these are basically, the neutrophils make these as a way to tunnel through the tissue. And so if you have too much neutrophils entering the tissue, secreting this MMP9, it can cause excess tissue damage. And we see this in rheumatoid arthritis. We can inhibit it by INF gamma, which is a Th1 response, and we get into that more into my other videos that I have created for the course. And this causes a lot of local inflammation. Now, let's talk about the memory. Once our T and B cells have made cells that's specific for a particular antigen from an initial exposure, it will always remember that antigen. So this is how it works, okay? So our first exposure of an antigen can go either of two ways. Number one, it can kill you, or it can be met by the innate immune cells, the neutrophils, macrophages, and dendritic cells. And they can kill the pathogen. But if their response isn't good enough, you may still die. Okay. Now, the innate immune cells then can present that antigen to the adaptive immune system, the T and B cells, and they will make specific immune cells to kill the pathogen. Okay, but then they're going to have some left over and make a memory. You know, they don't ever shrink back to zero. So once you have T and B cells made for an antigen, they will shrink way down, but they're never going to go to zero. So what happens with the second exposure? That comes in, the innate immune system, it acts first and it goes to kill the pathogen. It can also recruit the memory cells, which are very rapid now, because now it doesn't take days to make it, you know, just within hours. This can be ramped up really quick to snuff out an infection, and it will kill the pathogen. Okay, and so just remember that the memory cells are made from the T and B cells to help help a future infection. Now, the T cells are made from, depending on the antigen, there can be 4 different types of T helper cells produced. Okay, so the T cell is made in the bone as a naive T cell, and it matures in the thymus. So when an antigen is presented to the T cells in the lymph by an antigen presenting cell, such as a macrophage, dendritic, or B cell, it will give instructions through the cytokines that it produces to induce the naive T cell to mature into one of the types of T helper cells. And this is called polarization. There are 4 main polarizations of the T cells. There are a few others, but these are the ones that I discuss in my immune course that will launch this summer. The T cell polarizations can be assessed and managed naturally. So how do you change your immune system? So in functional medicine, we always want to remove triggers first as a foundation before adding anything. We want to clean the environment of toxins. So if you're living in a moldy environment, you got to remove yourself from that environment. You want to make sure your air is clean. You want to remove all of the inflammatory food and allergens from the diet. You want to replace the harmful chemical products with non-toxic and safe personal care and cleaning products to lower the total body load of toxins. You know, and then you start an anti-inflammatory diet for a few days and then start clearing any gut pathogens. So remember that the limbic system will send out a signal to the adrenals to release stress chemistry for anything perceived as a bodily threat. And we want to stress the importance of limbic retraining and restoring health. And, you know, and I the link in the Facebook thread for the limbic retraining courses that I recommend. They're excellent and they work really well. So next, what we want to do is repair the epithelial and mucus lining of the small intestines. This is also the time to fix any anemias that are discovered from blood work and improve tissue perfusion and blood glucose regulation to kind of quiet down that inflammasome activity. And then we restore. You know, that starts by removing triggers, being in a clean air environment, clean food, to reduce all of the inflammasome activation. I discussed that in a lot of and deep detail in the immune course that I'm launching. Now, the modulation of the immune system should be focused on taking the load off of the adaptive immune system and boosting the innate system to decrease the risk of autoimmunity from presentation of self-tissue to the T cells. So running labs on how the immune system is polarized and to rule out any obvious problems with anemias, blood glucose regulation, and oxygenation is key, and it must be reviewed first. So killing pathogens and fixing nutrient deficiencies can help reduce the inflammatory process, and targeted nutrient and herbal therapy to bring the immune system back into homeostasis. And this can be done in a very short time. So we can use functional foods and herbs and one of my favorite is polyphenols. They really help they're in flowers, leaves, and barks of vegetables and fruits and herbs have the ability to downregulate inflammation that's common in many of these chronic conditions and help quiet the inflammasome activity. And the ability to regulate this process is an important focus in functional medicine. These dietary phenols are not absorbed well, and they're available for metabolism by the colonic microbiota, which makes the metabolites that are further metabolized in the liver to several different compounds. And you can see that they are metabolized to anthocyanin, isoflavone, and all of these different flavonoids that are antioxidants and have a lot of immune regulation function. So using a plant-based polyphenol formula offers a potential new target to influence the T-cell cytokine profile, which may help prevent tissue damage commonly related to inflammation. So the way that polyphenols are metabolized, just some main points, they have difficulty in crossing the epithelial layer and the intestines, and typically have low blood levels and a high choline content. Then the colon microbiota will metabolize the polyphenols into varying compounds that can then enter the systemic circulation. That's difficult to measure because the colonic bioconversion of the polyphenols can vary greatly between individuals. And it also depends on the microbiota content, composition, as well as other foods present in the colon. Plus, they're metabolized in the liver, making intermediates that may have a different bioavailability. So they're very difficult to measure. And so the bottom line is, because of the vagaries and nuances of measuring cytokines, the symptom improvement may be a better marker than lab or cytokine values when measuring the impact of polyphenols on treatment. So, and also large quantities of polyphenols may be required, and extracts are significantly more concentrated than the dry herb and more efficacious, and combining several herbs can be synergistic. Oops, let me go. All right, so the polyphenols, so this is how they work in immune modulation. So they help with the, something happened to this slide here, but this is supposed to be the innate immune system. It helps decrease the neutrophil infiltration, decreases the MMP9, and improves circulation, and decreases the myeloid-derived suppressor cell activity. And with the adaptive immune system, it gets downregulated, which helps to decrease risk for autoimmunity, and it blocks the excess TGF-beta that causes fibrosis and scarring, and it helps to regulate T cells and mast cells and allergies, and it aids in detox, digestion. In the lipopolysaccharides, that's those nasty bacteria in your gut that makes those inflammatory things, and it's the gatekeeper of detoxification and works as a fantastic antioxidant. So when I was in my my doctorate program, we did a clinical study on the product that I had developed, ImmunoThrive, and which had the ingredients of Moringa oleifera, Mariah coingi, turmeric, spirulina, and pine bark, and what this formula was studied in 30-day IRB approved clinical trial through the University of Maryland Integrative Health. And this case series consisted of only three participants that were diagnosed with metabolic syndrome, which is a known inflammatory condition. They were given a symptom questionnaire known as a MSQ, and at the beginning and at the end of the study, the participants took 3 capsules daily of the extract for 30 days, and the MSQ scores had a considerable improvement. The laboratory findings were inconclusive, but as we just went through, it's kind of difficult to measure that in the lab. but the 3 participants, they all stated they noticed that they had more energy while taking the supplement, and there was a 67% overall symptom score in this study. So phenols can also be produced. Foods that are rich in polyphenols are the brightly colored fruits, the brightly colored vegetables, red wines, coffee, black and green teas, spices, and dark chocolate. Now, gut bacteria and yeast also make polyphenols. And some people have a difficulty in processing these polyphenol compounds and certain preservatives and food dyes and additives. And what happens is they become trapped in some part of a metabolic cycle, presumably involved with various detoxification pathways. So an individual with a phenol sensitivity may be intolerant to certain polyphenols. But most people can tolerate it well, but if they can't, it's usually, turmeric, fenugreek, oregano, thyme, rosemary, and occasionally immunothrive and it's getting trapped in the detox pathways. So this is the end of my immune webinar, and I'm going to offer a special of working with me. Okay, so if you want to know if this works, feel free to post a question to anyone that's worked with me in the Facebook group. I have a high level of success. Over 95% of the people that work with me have reported a significant improvement in their symptom scores. I do a comprehensive review of diet, history, exposures as they relate to your symptoms and the immune issues. So we can do an online remote through my secure online portal or by phone for a 1 hour long session face-to-face. So this offer is going to include labs through the direct-to-consumer lab, through Principal Labs, which is done by LabCorp, and that's a $250 value. So if you sign up for my consult, going over your labs and evaluating your immune system and your history, it's really going to save you over 50%, or actually, because I typically require an initial consult before I order any labs. So that's an additional consult that you can save. So the packages are available. The offer is going to expire the week. I'm going to post the link if you want to sign up in the Facebook group. If you're one of my clients already, you can send me a message through the portal. If you live in New York, they don't allow these direct-to-consumer labs, so it's not available. It's not reimbursable by insurance or Medicare, but you can use your HSA spending account for this. So what you get is a one-on-one, 1 hour long phone or video consult, value $450 for new clients or $300 for follow-ups. You get the bioscreen marker, which is 59 biomarkers, and a urinalysis, okay? The key areas covered in the panel is blood sugar, infection analysis, electrolyte analysis, anemia analysis, thyroid analysis, lipid status, liver and biliary, and a urinalysis. Now, these are all the really baseline things that we have to look at to see if, make sure that you don't have any kind of anemias, dysglycemias, problems with perfusion, biliary issues, liver issues that have to be addressed before we can start with modulating the immune system, okay? I will provide you with a written summary of our consult with a personalized protocol, and any supplements that I recommend, you'll be able to get them through my full script at 25% of manufacturer retail price. So you'll click on the link to register and pay, and then sign the informed consent and HIPAA and the policy and guidelines agreement. You'll receive an email from me that you have pending forms to fill out, and you'll need to fill out those forms within 72 hours so that we can get the lab work ordered, Okay, and I have to have information off of that to process your requisition first. I do have a 72-hour refund policy. So if you change your mind within the first 72 hours, you can get a full refund. However, after that, the lab portion of the $250 will not be refunded because I pay for those labs up front when I order them. And then you take your requisition to LabCorp for the blood draw. I prefer you do an overnight fast if you can. If not, then wait at least 4 hours after eating for the blood draw. So if for any reason you have difficulty with your blood draw, you can always contact me. So when the lab results are received, they'll be uploaded in your portal and you'll receive an automated email informing you that I've shared documents with you. So those results are yours to keep. Don't send me emails asking questions about your results, because when you get your results, what you need to do is click on bookings to schedule your consult with me. And that's the end. And I'm going to open this up for questions. Oh my gosh, we got a lot of stuff in the chat. Let me turn my video on so you can see me. Hi everyone, glad you all joined me. Let me see. Let me go here and look at the chat. Hi, Jeannie. Let me see. Okay, I'm coming down further. Does anybody have any questions for me? I don't see any questions. Oh, hold on. Is my chat working? Yeah. Okay. I can unmute participants when I ask me. Okay. Okay, I've unmuted. If please mute. Okay.
Oh my God, I can't mute everybody.
Okay, I had to mute everybody because everybody was just too loud. Anybody have any questions that you want to share and put in the chat? I don't see any questions in the chat. Somebody sent me a private message here. What does it say? Okay, so, okay. Here's all the chats. Now they're coming up. Would the evaluation work with Hashimoto's? Absolutely, yes. It does work with Hashimoto's. What is an example of a condition, a dysglycemic? Okay, so an example of a dysglycemic condition is if your blood glucose is greater than 100, then you have dysglycemia, okay? And let me see. Is there any supplements I have to go off of? For the test, no, okay? I just want you to be fasting. Would it work for Hashimoto's? Yes. A replay. The broadcast cut out often and didn't hear well. Yeah, I'm going to offer a replay later on in the week. And how do we sign up for this? I'm going to put a link in the, you know what, I can just put a link in the chat, actually, if, right here. Let me put a link here in the chat. I think that will, how come I got a private message? Oh, okay, here we go here. Let me do this. Okay, so that's the link if you want to sign up for the webinar special. So did y'all understand this pretty easy? Did you understand what I was explaining about the immune system? Do you understand your immune system a little bit better? about low glucose levels? Yeah, low glucose levels is a problem too, because then what happens is your brain's not getting enough energy. So your brain needs two things to work. It needs oxygen and it needs glucose. So when your brain is inflamed, it's using a lot more energy and it's sending off a lot of stress chemistry. So then you get reactive hypoglycemia. So if you have low blood glucose, we need to figure out what's going on that's causing your brain to be inflamed. That's how you fix the reactive hypoglycemia, is you find out the triggers, okay? Okay, the sound is muffled still. Having trouble with thyroid meds not being effective in chronic hypothyroid. Yeah, so the thyroid is the #1 organ in the body that's affected with inflammation and the immune cascade. So we have to figure out what the triggers are, all of those pillars. Okay, so somebody only saw the last 5 minutes. Oh, I'm sorry. Okay, so did y'all enjoy this? You can type in the chat. Okay, good deal. Okay, I enjoyed it too. And I'm gonna end the recording. I'm gonna stop the recording. You like the IgG explanations? Yeah, it is kind of difficult, and a layperson would kind of have to like get the transcript and study it along the slides and a little bit. But I think a typical layperson would be able to get it if you had the pictures and then the printout, the whole transcript, and study it. And you know, I think you'd do a whole lot on your own. Now, I don't have a printed resource of this. This is going to be part of my immune module that that's going to launch this summer with the toxic mold diet, the mold detox diet. We're having a whole practitioner module, and I made the whole immune for that. I've been working on it for a year. Okay, so yeah, to get the transcript, you would have to purchase the course. Okay. Do I have a mold live webinar available? No, I don't, but you know, Dr. Christensen's gonna be repeating the toxic mold this summer. So I'll be posting the link for the summit and this summer, and you guys can sign up for it and watch it free for that week. Yes, this is good for chronic fatigue, yes. Well, the course is made for practitioners, but I made it in a way to where I cut out a lot of the real technical stuff so that you can kind of get a really basic understanding. So what is the immune system doing? What are all of the things causing it to be that way? And what can I do to put it back? Okay, and that's what my whole course is focused on. You know, I had over 100 and something lectures in functional immunology postdoctorate. And so from that, I put together 5 45-minute videos to teach other people how, you know, on a less technical level, how to do this, okay? Yes, the supplement ImmunoThrive does help regulate T cells and detox. Yes. Very impressive work. I need to hear it a few times. Yeah, so this will be part of the mold detox diet for practitioners that will launch this summer. If you want to watch the whole 5-part series, that's going to launch this summer. And the ImmunoThrive, yes, that's the product that I developed. And if you want to try that, try ImmunoThrive. I was trying to make a coupon for the webinar, like a 20% off. And, you know, I'll post that in the Facebook group later when I figure out how to, Amazon's kind of tricky, whatever. Okay, so, let me see, Immunothrive would be next. How about to support liver for people with MTHFR and mold? Okay, so, you know, you want to, where you start as a layperson is cleaning up your products, cleaning up your air, cleaning up your environment, cleaning up your diet. And then, you know, the rest of the stuff is you have to remove all of those triggers. And, you know, if you want to work with me to look at your lab work and see how, what else I, if there's any other triggers and all of that. I'll be advertising about the launch of the summit this summer when it's available. Okay. So do you have a basic understanding of the immune system now? You understand about how we have the two arms, and then you have the extracellular immunity and the intracellular immunity, and they kind of go in the tissues and pathogen and get cleared. Anyway, so let me see. So the picture and diagrams help. Yeah, the pictures and the diagrams do help. So with mast cell, you know, you want to remove like triggers. The most important thing with mast cell is the limbic retraining. So, you know, when I was sick with mast cell, I thought, oh, well, with chronic exposure, you get more and more and more memory sitting on the mast cell. Well, that's not true. What's happened is the limbic system is up here, 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 and you're sending off stress chemistry, even at rest. Your limbic system is chronically activated, and it's like sending off stress chemistry, and it's knocking off those membrane-bound IgE, causing a mast cell with any type of, any kind of external stimuli. So what you have to do is you have to do limbic retraining, which is a huge time suck. Man, I'm still doing limbic retraining. You know, one of the things that I do is this alpha stem, and this helps a lot because it's kind of like limbic retraining, like EMDR, you know, when you're sleeping. And when you're sleeping, you go into REM sleep and your eyes do this. And that's how it processes the stress of the day. So when you have trauma or chronic activation of the limbic system, it's not processing it. So everything is a trauma and everything's knocking off the mast cells. So wearing this is called the alpha stem. And I wear it several times a day. I put these little thingies on my ear, and I turn it on, and it sends little tiny waves, and I can feel a little pinprick, tick, 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 tick. So it's doing this, and as soon as you put it on, it starts scrambling your brain waves, so your limbic system's not firing. So if I'm stressed or, you know, overworked or overtired, I put this on, and I wear it. So I typically wear it like 3 times a day, you know, and I just look really cool because I put it on my ears, and everybody thinks I'm like this cool grandma that's walking around with earbuds, you know, and my iPhone in my pocket. And, you know, so I wear it a lot. It's a patented technology, and it's only available through doctors. So if you're interested, you know, you can get it through me. The cost is $800, and I have like a rental deal. So if you bought it and you didn't like it, if you return it within the first 3 months and you say, oh, this isn't working for me, then you can get your, I'll refund your money less $100 for each month that you kept it, and shipping. So, you know, those are really great. And then the limbic retraining and neuroplasticity, the tapping, yoga, you know, laughter yoga, deep breathing, meditation, all of those things, and tapping, you know, that EFT tapping, that retrains the brain, and doing therapy, group therapy, and EMDR work with a therapist, that helps to retrain the limbic system. So for mast cell, that's the best place to start. You're not going to find any supplement that's going to fix that. You can find some supplements that help stabilize it, and Immunothrive is a good supplement that, you know, that worked for me in stabilizing my mast cells tremendously. But, you know, without the limbic training, you're going to be stuck with that mast cell activation for life. All right, guys. Well, it's been great, and I'm going to end the recording, stop the recording, and I'm going to